Empezamos.
Vale.
Okay. Well, good afternoon, everybody. Thank you for watching us. Welcome to the webinar organized by the Society of Spanish Researchers in South Africa and the Embassy of Spain in Pretoria. I'm Alberto Valenciano. I'm a member of the Society of Spanish Researchers in South Africa, and I'm going to introduce this webinar. Today, the Spanish researchers invited is Dr. Carmen Nacarino Meneses. She holds a bachelor degree in biology and a master's degree in biodiversity, both in the Universidad Autónoma de Madrid in Spain, and she did her PhD in paleontology at the Institut Català de Paleontologia Miquel Crusafont and in the Universidad Autónoma of Barcelona, both in Spain. She is currently a postdoctoral fellow in the Department of Biological Science at the University of Cape Town in South Africa, and she receives funding from the Center of Excellence in Palaeosciences. Her research focuses on the paleohistological study of different groups of mammals, with a special emphasis on horses, zebras, and their extinct relatives. She analyzed their bones and teeth using histology, which means using a small fragment of bones under the microscope, but I'm sure she's going to explain much better than I, in order to obtain different information about the paleobiology, paleoecology, and evolution of these forms. So, of course, you can leave any questions to Carmen in the chat of this Zoom, and at the end, I will address this after the conference. Before to listening and watching this very interesting conference, because I know Carmen and she's a very good speaker, we are going to see a small introduction of the ambassador of Spain, Don Carlos Enrique Fernandez Arias, which he is going to explain a little bit about this. So let me share this. Okay, this is a whale. Okay.
Good evening, and thank you for joining us once again. Today's webinar, Inside Bones and Teeth, Deciphering the Biology of Extinct Animals Under the Microscope, is presented by Dr. Carmen Nacarino Meneses, Postdoctoral Fellow in Palaeohistology, Palaeobiology, and Paleontology Department of Biological Science of the University of Cape Town. She is going to explain to us the importance of Palaeohistology. It means the study of fossil bones and teeth under the microscope. It provides information about key research topics, from the reconstruction of biological characteristic of prehistoric animals, for instance, the age of death, to the evaluation of the process of formation of archaeological and fossil remains, diagenesis. Palaeohistology can tell us about how fast or slow extinct animals grew, at what age they reproduce, and how long they live. I hope they interest you. Enjoy it. Thank you very much.
Thank you so much, Carlos. So, sorry for this. Carmen, the floor is yours. It's your turn.
Thank you so much, Alberto. Okay, so let me share my screen. Okay. Okay, I think everyone is seeing my screen. Well, so first of all, thank you so much, all of you, for joining us today in this webinar. I would also like to thank Alberto for his introduction and the Ambassador of Spain, Don Carlos Fernandez Arias, for his very kind words. First of all, I would also like to thank the Embassy of Spain here in South Africa and the Society of Spanish Researchers in South Africa for inviting me and talk today about my research. So, as both the ambassador of Spain and Alberto said, my area of research is palaeohistology. But what is palaeohistology? So, this very long and kind of weird work in English came from three different works in Greek, which means ancient, tissue, and study. So, we can say that palaeohistology is the study of fossil bones and teeth under the microscope. And by microscope, I mean every kind of microscopy that you probably know, from the scanning electron microscopy that allow you to see very tiny details of the bones and teeth, to transmitted light microscopy, which is the common microscopy that most of you have used probably during your years at the high school or university, and to polarized light microscopy, which is the one that we usually use when we study fossils. This kind of microscopy is very similar to the transmitted light microscopy, but these microscopes incorporate two different filters that make the bone tissue and the dental tissue to show these colors that help us to identify different features. So regardless of the kind of microscopy that you are going to use in your paleohistological studies, you usually need to prepare the samples. Specifically in my case, I need to prepare what we call histological slides that are very thin sections of bones or teeth. And how do we do that? Well, the first thing that you have to do is take measurements and photographs of the sample, and then you embed it in an epoxy resin, the whole sample or part of it, and then you cut the piece that you are going to study. Then you grind this block using a grinding machine, and you also polish it to eliminate any cut surface. And then you mount the interest surface into a frosted glass. And this mounted specimen on the frosted glass is later cut again using a specific machine that allow you to cut very thin sections of samples. So here, for example, you see one slide of around 500 microns. But this slide is still too thick to be observed under the microscope, so you still need to grind it a bit more, both using the same machine that you use it to cut it, that it automatically grind the sample. And you can also grind it a bit more by hand using another machine, just to get the final thickness that you need to observe the sample under the microscope. So once you have this, you are ready to, as I said, observe your sample under the microscope. So you have your bone, for example, you prepare your slide, and if you are lucky, you will get something like this. And what are we seeing here? Well, we are seeing the place that vascular canals occupied when the animal was alive. We can also identify the spaces occupied by the bone cells when the animal was alive. These are these small black dots all around the image. You can also identify how the different collagen fibers were arranged when the animal was alive. These are the small lines that you see also all around the image. And moreover, you can also identify different features of the bone, which are called primary osteons and secondary osteons. In the case of teeth, when we prepare histological thin sections, we don't see the same at first that we see when we prepare bones, because teeth are formed by three different tissues. These tissues are the enamel, the dentin, and the cementum. And when we observe them more in detail, we can see that all these tissues also present several features. In all of them, we can identify several lines that are marked here with arrows that are called incremental lines. In the case of enamel and dentin, for example, you can also identify other structures. And in the case of cementum, you can also see the places that cementum cells used to be, these small black dots. So at this point of the talk, some of you maybe are thinking, okay, you are showing us very beautiful pictures, but is it really worth it to partially or sometimes completely destroy a fossil bone or tooth, which is unique and irreplaceable, just to study their histology? So, well, I must say yes, and I'm not biased at all. But no, really, it's yes, it is completely worth it to do that, because by studying paleohistology, we can get information about so many different things. For example, we can get information about the process of diagenesis, which is the process of formation of fossils. Here, for example, you have two different images of bone. In the top one, you can see a lot of dark areas in which that doesn't allow you to recognize the bone tissue properly. And these black areas are formed as a result of the action of bacteria during the decomposition of the bone. So because we know that this bone was affected by bacteria, we can say that it experienced variations in moisture during diagenesis. On the opposite side, we have the other image in which you can identify very well the different features that I told you before, the vascular canals or the osteons. So in this case, we know that this bone was not affected by bacteria. And it was because it was probably buried in an anoxic waterlogged soil. But we can also get information, for example, about pathologies. Here you have the image of a bone from an extinct seal. And here the authors identify some area that look quite strange for them. And by using bone histology, they could say that this area was an osteosarcoma, which, as you know, is a kind of bone cancer. But using bone histology, well, dental histology also, we can also get information about life history. This is my main area of research, and this is what I will tell you about during this presentation. So for those of you that never heard about life history before, I can tell you that life history is the combination of developmental stages and demographic events that an organism experiences during its life from birth to death. So here, for example, you have an schematic representation of the life cycle of the Asiatic wild ass, the Equus hemionus. And you can see that this animal, as all other organisms, experience different stages. So all this life cycle and how the animal lived that life cycle is what we understand as life history. And each of these different stages is what we call life history traits. So life history traits can be defined as the demographic characters and the covariant biological features that affect survival and reproduction. In mammals, for example, some of the most important life history traits are the size at birth, the age at weaning, the adult body size, the length of the pregnancy, the age at first reproduction, the number of offspring, or the interbirth interval. And the cool thing with bone histology is that it registers some of these life history traits. So for example, by studying bone histology, we can get information about growth rate, age at maturity, or age at death. Estimations about the growth rate are mainly based on the study of bone tissue types, while estimations about age at maturity and age at death can be obtained by means of a methodology known as skeletal chronology. So the relationship between bone tissue types and inferences on growth rate is based on the statements of what we call the Amprinous Rule. According to this rule, the different bone tissue types are formed at different rates. So for example, the lamellar bone, which is a bone tissue that presents a bone matrix very organized, is formed at very slow rates. On the other hand, we have the fibrolamellar bone, in which the bone matrix is less organized, and then it is formed at a faster rate. In between these two ends, we can find other different tissue types, such as, for example, another one called parallel fiber bone. So the thing is that this close relationship between bone formation and growth rate has led some authors to use the identification of different tissue types to infer differences in the rate of growth between extinct species. This is, for example, what these authors did in these extinct animals, Miotragus balearicus and Gazella borbonica. They studied their bone histology, and they found that the bones of Miotragus balearicus, for example, present a very lamellar bone tissue, while that of Gazella borbonica were formed by fibrolamellar bone. So because lamellar bone was formed at slower rates than fibrolamellar bone, the authors concluded that Miotragus balearicus grew at slower rates than Gazella borbonica. But interestingly, bone tissue types do not only differ between species, but also during ontogeny. This means that young individuals present different bone tissue types than old individuals. So, for example, in mammals, it is common to find that the bones of young organisms are composed of fibrolamellar bone. When the animals grow and attain their adult body size, they started to form a slow-growing lamellar bone on the periphery of the bones, which we usually call external fundamental system or outer circumferential layer. So actually, the transition from this fibrolamellar bone to the lamellar one is thought to mark the attainment of maturity. When we observe the fossil bones under the microscope, we do not only see bone tissue types and osteons, but we also identify other features that we call bone growth marks. These can be lines of arrested growth, and as you have on the image on the left, these are very thin dark lines that appear in bone cross-section and that represent moments of cessation of growth. Bone growth marks can also take the form of annulus, as the image on the right. In this case, these are poorly vascularized rings of lamellar bone that do not represent a stop in the rate of bone formation, but just a decrease in the rate that the bone is being formed. So most of the bone growth marks that appear within a bone cortex are deposited annually in response to metabolic and hormonal cycles. Several years ago, for example, a study showed that these cyclical bone growth marks are formed during the winter season. During this season, during winter, the animals that they studied that belong both from the northern and southern hemispheres. So these animals experience a series of changes in their metabolism and in the concentration of several hormones that impacts on the rate of bone growth and that lead to the formation of these growth marks. So as we know that these bone growth marks are formed annually, we can count them and we can get information about different life history traits, which include the age at death, the age at maturity, and the growth rate. So, for example, if we count the total number of cyclical growth marks that we found within a bone cross-section, we can get or we can obtain an inference of the age at death of this animal. We can also get information about the age at maturity by counting the number of cyclical growth marks before the appearance of this slow growing tissue that I told you that marks the attainment of maturity, the external fundamental system or outer circumferential layer. And finally, we can also take measurements in these cyclical growth marks. We can measure, for example, their perimeter, and we can perform plots like these ones to see how the different bones grew and to try to identify differences between extant and extinct species, for example. However, not all cyclical growth marks within a bone cortex are deposited annually. Some of these growth marks can also appear during moments or biological events that suppose a moment of physiological stress in the organism. These are the so-called non-cyclical bone growth marks and can appear, for example, during the metamorphosis, the hatching, or the weaning. Here in these images, for example, you have a non-cyclical bone growth mark that we identified in the different limb bones of baby zebra, and that we think that it represents the moment of birth, that it was formed during the moment of birth. So not only bones provide information about life history, also teeth record a lot of information. Specifically, when I study teeth, we usually get information about growth rate, age at death, age at maturity, and age at weaning. In the case of teeth, we can get all this information by analyzing the incremental lines that I told you about at the beginning of this presentation. Because these incremental lines also present a specific periodicity, similarly as the cyclical growth marks in the bones. So in the enamel and dentin, for example, incremental lines are formed on a daily or supradaily basis, while in the cementum, these lines are deposited annually. So if we focus, for example, in the enamel, and we take measurements of the daily lines, different measurements, and we calculate things, we can get different parameters, such as the daily secretion rate or the enamel extension rate, that inform us about how the teeth grow. Moreover, some of these parameters, such as, for example, the enamel extension rate, are considered to be a proxy of the organism's growth rate. So we can measure, for example, the enamel secretion rate in extinct animals and compare it with extant one to see if there's differences in growth. So here you have a plot that shows the rate of enamel extension for the three-toed horses from the Miocene and extant equus. And as you see, the enamel extension rate of extinct horses was lower than that of extant zebras. So this suggests that the extinct horses grew at slower rates than extant horses. But we can also count the number of daily or supradaily lines within a tooth's crown or within the complete tooth to calculate the time of crown formation or the time of tooth formation. This is interesting because if the tooth belongs to an immature individual, this estimation of the tooth formation time gives us the age at death of this individual or animal that we are studying. But also the interesting thing about estimating crown formation times is that in many mammals, including several ungulates and equids, for example, the time of crown formation correlates with the time of eruption of the tooth. And this time of eruption of the tooth also correlates with the age at at the age at several life history traits. So, for example, the eruption of the first and third lower molar correlates with the age at weaning and the age at maturity. So what we can do is we can estimate the time of crown formation of the first and third lower molar in an extinct species. And doing that, we can get an inference of the age at weaning and the age at skeletal maturity for this species. And this is, for example, what these authors did in the bovid that I speak about some slides ago, Myotragus balearicus, and compare it with extant sieves. So they observed that the third lower molar takes much more time to be formed in Myotragus balearicus than in extant sieves, which suggests that maturity was delayed for this Balearic species. So all these measurements and observations and these kind of things on bone and dental histology is what I did for my PhD in Spain. As Alberto said, I focus most of my research in studying different extant zebras and one wild ass. And I also applied later this information to several Pleistocene species from different parts of Europe. But when I finished my PhD, I decided to move to South Africa. And maybe some of you are thinking or are wondering, and why South Africa? especially my mom is wondering, why South Africa? It's very far away. Well, I moved to South Africa because I wanted to join the Palaeobiological Research Group at the Department of Biological Sciences at the University of Cape Town, because this group has a very extensive experience on the analysis of bone histology in many different groups of vertebrates, including dinosaurs and early mammals, among others. So I was lucky that I got a postdoctoral fellowship from the Center of Excellence in Palaeosciences, and I could move to South Africa and learn with them. So what am I doing here? Well, as a biologist here in South Africa, I am trying to enjoy the much that I could the outside. I am trying to enjoy the wildlife, the hikes, the nature, everything. But I think my supervisor is also watching this presentation, so I must say that I'm also working very, very, very hard. And especially, I came here to South Africa to study the bone and dental histology, of course, of two different extinct horses. One is Nathoipus Hoyeri, the three-toed horse from the early Pliocene site of Langebambeek, and the other one is Equus capensis, which is an extinct zebra from the Middle Pleistocene. Specifically, my research will focus on the fossil site of Elandsfontein. So now I'm going to show you the first results that I got by studying the bone histology of Nathoipus hoyeri from Langebambeek. And if you have attended other seminars of the Society of Spanish Researchers in South Africa, maybe you remember the talk from my colleague Alberto Valenciano, which is moderating this webinar, about this unique fossil site, Langebambeek. If you didn't attend it, I recommend you to go to the YouTube site of the Society of Spanish Researchers in South Africa and check it, because you will learn a lot about this unique place. But in the meanwhile, just in case you didn't attended this webinar, I can tell you that Langebambeek is a fossil site located on the outside of Cape Town, around 120 kilometers away of Cape Town, in which what we know now as the West Coast Fossil Park. So there, the bones appear, well, bones and teeth, the fossils, appear forming extensive bone beds. You can see here that there are a lot, but it's a high amount of fossils there. And the latest datations indicate that these bone beds belong to the Barsvater Formation and were dated around 5.2 million years ago. So Langebambeek is unique in many aspects, but specifically it's unique in terms of its biodiversity, because there has been described around 300 species of invertebrates and vertebrates. This includes many lower vertebrates, several kind of birds, and around 14 orders of mammals. 1/3 of these mammals are carnivores, and there's around 17 families of herbivores. Among these herbivores, it is noteworthy the presence of a three-toed horse that is named Nathoipus hoyeri. I will try not to say this name a lot of times because it's very long and very difficult to pronounce, and I'm going to pronounce it but at some point. But this is the name. And this species was described based on an adult female skull that present a mix of primitive and advanced characteristics. The authors that described this species also said that it presented very slender metapodia that suggests that it was, or it present an advanced cursoriality. It also presents very high crowns, which it also presents very high crowns, which, along with some dietary analysis on the microware of this sample, suggests that it was a grazer which had a diet similar to that of extant zebras. And probably this animal occupied a similar niche than extant equus. So now we have a very good knowledge of the morphology and the paleodiet of this animal, but there's still much we do not know about its biology. And there is where my research came, because what I'm trying to do is to infer some aspects of its paleobiology using bone histology. So I started by studying 4 metapodia that belongs to the collections of the Ithaca Museums of South Africa. And I prepared histological slides following the procedures that I already showed you in the video at the beginning of the presentation. And I later studied these thin sections using polarized light. I also took several measurements to infer the body mass of this species. And here is what I saw when I put the slide under the microscope. This is the cross-section obtained from a metacarpus, and on a close-up of this image, you can see that the main tissue that we found is a fibrolamellar bone, so a very fast-growing tissue that present a lot of longitudinal, circumferential, and radial vascular canals. We also identify the outer circumferential layer, so the slow-growing tissue on the outside of the bone. In the metatarsus, we see the same tissue. We see a highly vascularized fibrolamellar bone that presents multiple longitudinal, radial, and circumferential vascular canals. We also identified an outer circumferential layer. So we can say that the histology of both bones is practically the same, or is at least very similar. But the thing is that when we compared our results with that reported for other Hipparionids from Europe, we observed that our bones, so the bones of the Lange-Van horse, were much more vascularized than in other Hipparionids. And this suggests that Nathoipus hoyeri presented higher rates of growth. This is probably related to its larger body size, because as you can see here, we estimated an adult body size of around 215 kilograms for this horse from Lange-Van, while European Hipparionids only weighed around 200 kilograms or a bit less. But we did not only study bone tissue types, we also studied cyclical growth marks. Specifically, we found 3 cyclical growth marks in both the metacarpus and the metatarsus of Nathoipus hoyeri before the presence of the outer circumferential layer. Because these cyclical growth marks are formed annually, and the presence of the outer circumferential layer indicates the end of bone growth, we can say that metapodia of this animal finish their growth after their 3rd year of life. And again, when we compared our results with those described for other Hipparionids from Europe, we observed that European Hipparionids present only 2 cyclical growth marks before the outer circumferential layer. So this indicates that the outer circumferential layer appears later in the African horse than in European Hipparionids, and probably that the age at the skeletal maturity is delayed in this in the Lange-Van horse as compared to the European Hipparionids. Along with cyclical growth marks, we also identified a non-cyclical growth mark that represent the moment of birth. So the same mark that I told you about in previous slides. So we measured its perimeter, and we compared it with the perimeter of this line in both other Hipparionids and extinct equus. And our results show that the perimeter of this neonatal line is larger in Nathoipus hoyeri, so in the Lange-Van horse, than in European Hipparionids. The neonatal line, however, or the perimeter of the neonatal line is smaller in Lange-Van-Beck's horse than in extant and extinct equus, which are larger, present also a larger adult body size. So our results on the neonatal line agrees with allometric predictions. So this first approach to the bone histology of Nathoipus Hoyeri from Lange-Van-Beck revealed several information about its paleobiology. On the one hand, we found a more vascularized fibrolamellar bone in Nathoipus hoyeri than in medium and large size Eparionines from Europe, which suggests that it presented higher rates of growth. We also identified more cycles of growth before the deposition of the outer circumferential layer, which indicates that its age at maturity was probably delayed. And finally, we found that the perimeters of the neonatal line were larger in the African Eparionine as compared to medium and large size Eparionines from Europe, which indicates larger size at birth. So all these results were the expected given the larger size of Nathoipus hoyeri as compared to late Miocene equids from Europe. And this is all. Thank you so much for your attention. I would like to thank again the Embassy of Spain in Pretoria and the Society of Spanish Researchers in South Africa for giving me the opportunity to talk today about my research, and also the Center of Excellence in Palaeosciences for founding my research. And of course, thank you to all of you for watching this webinar. Thank you. If you have any question, I will be happy to answer it right now.
Thank you so much, Carmen. It was a very nice presentation. And I have to say that I love the video from the beginning. Because it was
Thank you.
very, yeah. So, have we some questions? We have 2 questions. So I'm going to, please, if you have any question, write both in the chat or in the Q&A section. Okay. Yes, Mario Mairal have 2 questions. The first one is, how far does the ISU technique work? Million years?
Yes. Actually, most of the histological works done so far are done in dinosaurs. So not all of them, of course. There's a lot of histological work, especially during these past years. But yeah, it doesn't matter that the bone or the fossil is younger or, I mean, yeah, from recent or older. You can use both bone or dental histology and analyze them. So there's a lot of works in dinosaurs, for example. Sometimes you can get the problem. I said at some point of the presentation, I said that you will get something like this if you are lucky, because sometimes the problem that you have is that the preservation or the micropreservation, I would say, is bad. So when you look at the bone or the teeth from the outside, it looks completely fine, like another fossil or a normal bone or teeth. But then when you cut it and you observe it under the microscope, you find these black areas that I told you also in another presentation, which are the result of the action of bacteria. And therefore, in these cases, you can't study the histology. But it's not a question of how many years this was underneath the earth or anything. So
yeah. So having a fossil of a bone doesn't mean you have.
No.
You can. Yeah. It's dependent of the preservation.
Yeah.
Okay. Thank you so much, Mario. Mario have another question. Do histological evidence correlate with climate? Sorry for my English. If I didn't say that at the beginning, I have to say that. Climate fluctuations or give any information about this? So yeah. So I think he means that if you can obtain information about the climate, like you have in an analogous way with the 3.
Well, the idea of skeletal chronology is based on dendrochronology from the trees I know why you asked that, because you work with trees and plants and these things that I know you.
Wait,
But
you mean Mario, not me.
Mario,
Sorry.
Mario, yeah. So it's there's still much work to do to completely understand why the lines of arrested growth are formed. Okay, I have to say that at first. We get a good idea that there's a lot of mechanisms around relating the metabolism and the hormones, as I said before. So there's, climate is influencing the formation of these marks, but we still do not know well how. Of course, climate can affect the metabolism, and therefore it can affect at some point the formation of marks. They can also affect, for example, I think that imagine that there's a change in climate, so the animals had to reduce the rate of growth. And maybe we don't see a different bone tissue type, but maybe we see that the growth marks are closer to each other. So this can also give us information. I hope that this answer you more or less.
Thank you, Carmen, and thank you, Mario. Please don't be shy. If anyone have any questions, please just write them. We have more questions. Okay. So, this is very long. Ayan, thank you. Say, how reliable is using growth lines in determine the age of death of adult bones? And you can read the rest of the question.
Okay. Thank you for this question, because you are right. This thing, he said that I was taught at university that mammal bones grow by solution and replacement, which I would have thought might obscure this. This was a long time ago, so I expect these techniques have progressed since. So yeah, you are right about that, and let me explain this. This was a very or I try to make it very easy. So I just say, yeah, you just count the number of cyclical growth marks within a bone, and that's all. You can get the age at death. But as you say here, it's not that easy. On the one hand, there's this thing. There's the secondary formation of bone, which you said that the fibrolamellar bone, for example, that I told you that most young individuals have, during life, this bone is resorbed and reformed again. So, of course, and therefore it appears what we call secondary osteons. So these secondary osteons can erase previous bone growth marks. And not only that, but also the medullary cavity, the part of the inside of the bone, also changes through life. And in mammals, for example, this medullary cavity expands during life. So it can also erase some previous growth marks. In this case, what we try to do is to use ontogenetic series of bones. And of course, there can be some variation, but more or less, I don't know, a 1-year-old zebra more or less is always the same size. So if you use an ontogenetic series, you can superimpose the different bones and know more or less if any growth mark was loose because of secondary bone formation or because of the resorption of the medullary cavity. So yeah, there are some techniques, let's say, that can help you to know if there are some growth lines that got lost because of these processes that occur through life or not. But we still have these problems. And I'm glad that you asked, because I didn't have time to talk about that on the presentation.
Thank you so much, Ayan. I have a question. That is mine.
Okay.
So I see that for me, as a vertebrate paleontology working on taxonomy, it's very interesting that you obtain different histology in the horse from Langevanberg. Euratoipus or something like that.
Eurygnathoipus.
Eurygnathoipus, which is different to another genus of a very similar horse, which is Hipparion. So in that time, you have different taxonomy, different histology, and also they are different genus. Or
yeah, well, most of the Hipparions that has been studied histologically also belong to the Miocene, and Eurygnathoipus is Pliocene. So it's not only that there's a different taxonomy, but there's also a different age. There are probably so many different things that are affecting here. So yeah, it would be interesting to analyze other Eurygnathoipus members. There are some specimens from, I think, Kenya and Ethiopia, if I'm correct. Because actually Eurygnathoipus is a genus that appears here in Africa. So yeah, it could be interesting to compare them. I don't remember if these other Eurygnathoipus specimens are also Pliocene or Pleistocene, or I don't remember well, but it could be interesting to check it and compare.
yeah, sure. Thank you. Okay, we have another question of OCM.
Okay.
He or she say, thanks for this great presentation, Carmen. What about using microcity? Could we observe the same as you usually see with these thin sections?
Well, I have not worked a lot with microcity, so I can't tell you my direct experience because I have never checked this thing with the microcity, but I've read about it and I've seen other presentations from colleagues. And it depends on the resolution. The problem, or the main problem with microcity, is that we do not get the same resolution, at least as far as I know. I hope that at some point we can get a very good resolution of microcity, because in this case, we won't need to cut the bones and teeth. So it will help a lot. But for now, my experience is that there's not enough resolution, for example, to see these lines. You can see more or less the osteons and these structures, but it's very difficult to identify the lines. And in the case of teeth, for example, I think it's impossible to identify especially the daily lines. Maybe the supradaily lines could be seen with microcity, but not yet.
Not even with the synchrotron, right?
Well, with the synchrotron, we get much better results. And actually, with the synchrotron, there are studies. Synchrotron. Well, this thing. There are some studies with this, but this has a very good resolution. So in this case, yes, you can, but it's a very expensive technique. And this is why some people still do palaeohistology, because it's not as expensive as synchrotron.
Yeah, I understand. Thank you so much, OCM, and thank you, Carmen. Let's see, we have more questions. You have a lot of ratings for people.
Thank you so much to everyone, really, for attending and your comments.
Yeah. So, anyone have any specific question? No? Yeah, we got another one.
Okay.
Okay. Thank you, Bailey. Mark Weiss, sorry for my pronunciation again. Thank you for the awesome talk, Carmen. What did you use metacarpal and metatarsal? Well, he's saying in plural, and no other limb bones, such the humero or the femur.
Well, just because there are more. Usually when you go to the collections, especially in the case of horses that are very big animals, you can find lots of metapodial. I mean, well, lots, sometimes 3 or 4 boxes. But if you look for femora or humeri, sometimes there are 2 or 3 only. So when you apply for permission to cut, most of times they said, I'm sorry, but you can't cut that because we only have 3, and I understand it. I mean, if they only have 3, it's normal that they don't allow you to cut them. So this is why I use metacarpi and metatarsi. We did a work using extant specimens, and we saw that the number of lines is similar in the metacarpi as in the femur, for example. So we think that it can work. It will be better to use another long bones. And actually, during my postdoc, I also had permission to cut the femora of the Lange-Van horse. I didn't show here the results, but they are in agreement with the results of the metapodia. So it's just a question of numbers, nothing else.
Okay, thank you for the acceleration. Okay, I don't know. It seems more questions.
I see
Come
here
on.
someone with the hand raising. I don't know.
Where? Oh, I didn't see. Sorry.
If we have to do something or how this works. You can say allow to talk maybe and see.
Maybe caller have to. Okay. And what happened now?
I don't know. It's mute. So.
Solicitar.
I don't know. Maybe it was just.
Make it.
Well, maybe it was just.
You want to say something, make it, please. Write in the chat because I don't know how to work this software. Okay, we have more. Thanks. Fantastic presentation. I think it's the best histology lecture I have ever been.
Well, thank you so much.
But
I'm sure there are a lot of very good lectures. So, but I'm glad that all of you like it and I hope learned.
Yeah, I mean, for me, it was very instructive. I don't know.
Well, I like that you all like it.
Well, if anyone have more questions, I think we can say.
Yeah, you can always write me. You probably can find my email on the web. You can write me. You can also find me in Twitter. So just write me if you have any other question. I will be happy to answer you. Tell me.
I forgot to say that this webinar is recording. So in the future, it's going to be available in the web, in the channel of the Spanish Society of Research in South Africa. So yeah. Thank you so much.
Thank you so much.
Take care, everyone. Bye-bye.